Do you dare to give a timeline of when you hope to see gene therapies in patients curing heart diseases?
This is the date. So if you are entering the first clinical trial, then you just think about what we all had to experience in the last couple years with Corona. This is certainly something that could scare people.
Yeah. How did your pitches change between call 1 and call 60?
I think they became shorter.
Would you advise incubating it within academia itself, or would you advise kind of like spinning the idea out early? Welcome back. Today, we confront the future of medicine. Our focus, gene therapy and the quest to cure heart disease. Our guest exists in two powerful worlds at once. He leads a major academic division here at the University of Heidelberg as the W3 Professor of Molecular and Translational Cardiology. But that's just half the story. He is also the founder of Aavigen, a biotech startup that is rapidly converting lab breakthroughs into real-life cures. Join us as we explore the tightrope walk between scientific discovery and commercial development, and what it really takes to launch the next generation of life-saving medical technology. Please welcome Professor Dr. Patrick Most.
Thanks a lot for having me, and thanks a lot for the interest actually in our work. Let's see how this unfolds and if I can actually explain what we are doing and where we are actually heading.
Absolutely. Yeah. What first kind of pushed you to think of gene therapy as a viable solution for heart disease in the first place?
Yeah, so maybe let me start maybe with the remark that this is actually really a team effort. So it's nice that you're actually going to feature what we are doing, or maybe bringing me to forward. But this is, as I said, it's really a team effort. And so in terms of gene therapy, I think it is the work that I was privileged to do already during my doctoral thesis. So actually that dates back to the 90s. At that time, we were lucky to get in contact with a colleague from the United States, Professor Walter Koch, who actually also became my US mentor for later career building. So he was spearheading the gene therapy area at that time in terms of cardiac gene therapy. And we were lucky to get in touch with him. We were able to work with him at Duke University, and through that we had access to the gene therapy space. And then the topic of my thesis, which is about a certain molecule in the heart who has the ability to enhance contractile performance, amongst other things. Bringing these two things together, actually, that made the concept. And since then, we stayed on this task. And then moving it forward from the academic development, actually, as a necessity at the very end into the bioindustrial space where we are now today.
How does the transition work when you start in academia and going into biotech? What is necessary to make that happen?
Quite a bit. So this is not, I would say, as straightforward as it might look from today if you basically look in the rear mirror, right? I think a lot of these things were not planned at that point. I think if you wrap up the time a little bit, so from our very first understanding that this particular molecule, it's called S100A1, it's quite a fancy name, has these certain abilities in the heart. It took a while then to create all these experimental heart failure models that you need. But once we had our proof of concept that it therapeutically works, and in the best case scenario, you have this in a human relevant model, which also would include the size of a human. In our case, these are more or less domestic pigs. So there now we are talking about roughly 15 years of research from the very beginning to this final concept. At that stage, I think we had the data that convinced ourselves that the concept is robust enough that it could be taken forward. And I think if we now date back, and this is roughly 2011 when we had this kind of key publication that we got out, still it took us quite some years to convince ourselves that we could make this move. Because as you can imagine, now I certainly would like to mention some other people who were absolutely key and still are key to that. So first of all, that's Professor Sven Pleger. Today, he was my first doctoral student, so I think he was, I would say, the key connector to doing the work at Duke University. And then Andrew Rempes, that was my group leader at that day when I did my doctoral thesis with him. So he was also very ambitious and gave me basically the freedom and the liberty to do all the work then that I was able to do. And Professor Hugo Kartus, who is also a co-founder of the first startup and the second startup that we are today, who had been my clinical mentor and supervisor throughout my clinical career, and still now is a very, I'd say, key figure in my life, and also working with him closely together in the startup. So at that point, I think we basically understood that we have the data, but there was no funding available actually to translate it further, for example, into kind of a product that would be eligible to bring it to the clinic, despite the fact that we had actually not really a very good understanding of what we would need to do as further steps. So what we did at that time, we reached out to our technology transfer here at Heidelberg University. And so now we are talking about 2011, 2012. At that time, I would say the whole startup concept was extremely young. So our technology transfer got us in touch with the Mannheim Business School and with the department of Professor Voivode, actually. We were able to assemble a team of young students who helped us to build a business plan. And the business plan is some of the things that is for sure key, because it forces you to bring all the various aspects together that you need to overthink very closely if you would actually like to set out and found a company. That was completely new for me. And so I had a very ambitious team of young students at 7 who did her master thesis with this kind of business plan. So then for almost a year, I drove every Friday to Mannheim. There was a cafe, I think it's not existing anymore, against close to the Schneckenhof, basically where the business school is. So we've met there then for almost a year for 2 hours. And then they explained to me what they would need to know from my side to build a business plan, and vice versa. I understood much better what it would take economically, basically, to build this plan. So that was the first step. But in a second move, and that was also very important, a good friend of mine who is also now co-founder of Aavigen and who was also key for founding the first startup, that's Professor Mark Lerchenmüller. He's currently at Mannheim Business School, holding a professorship there. At that time, he was still with Boston Consulting Group, so he got a very, I would say, substantial training in bringing those things together. And so with him together, we further compressed or concentrated the business plan into a manner that was really suitable to set out and then basically ask investors maybe to fund this endeavor. And at that point, it was also clear that within the academic system, it would not really be possible to do this. So the German Research Foundation at that time, they didn't have any grants to fund work like this. Although I think translation was really becoming kind of a buzzword that was going around. But nevertheless, so I think that was difficult. And it was clear from the very beginning that the amounts that we would need would by far exceed public funding opportunities. Mm-hmm. So we took the business plan at that stage, which I think then was already quite decent. Then we asked ourselves the question, so how do we get in touch with investors, which is a completely different world, as you can imagine. And we made a decision, and in as much that we hired an interim CEO, Chief Executive Officer, and I think you might have heard the name already, or maybe recall it, Claudia Ulbrich. Mm-hmm. And so I've met Claudia in Berlin, and we discussed the potential opportunities that might be on the table, and then Claudia made the decision and came on board. So she became our interim CEO. And from then on, we used the various ventures that you could attend, and I think had numerous calls and interaction with VCs, and it was quite frustrating because I think after contact number 60, we realized that gene therapy is really not within the thinking, I would say, of the investors' world or understanding at that point. And then there was kind of a key call that happened, and that was to Forbion, which is one of the biggest European biotech investment funds, who is operating also on the other side of the Atlantic. And within, I say, a millisecond, you could understand that on the other side, the person that's Sander de Venter, Professor de Venter, immediately understood what this program could mean. And so after 3 or 4 minutes, normally at that point I was already trained in bringing up this whole story, and then you start to talk, and he very quickly interrupted me and said, look, if this is okay, I would forward the business plan to Unicure, and at that time Jörn Aldag was their chief executive officer. If that would be okay, and actually yes, of course. So would be quite happy if he would do that. That happened, and then I know within a week or so we got in touch with Unicure, and from then on I think we were on the road towards this, I would say, opportunity that we were looking for, maybe not really consciously, because at that point we also had to understand what's ahead of us, but it was, I would say, a very important lesson until then. And this is also what we have today, right? So today with our second startup, we also constantly are out there talking to investors, and you very quickly understand if someone is looking for this topic or not, and so you can deal much better with the time that you have to put in and have constructive conversations, but that might not really have a follow-up.
Yeah.
How did your pitches change between call 1 and call 60?
I think they became shorter because you understand what's important and what's not. But as I said, I think you realize within minutes if you have someone on the other side who is maybe an expert or who is actually looking for this investment opportunity. So I think we just had a conversation a couple of weeks ago back, and I think someone was saying, yes, you will not really convince an investor during a meeting. So either they are already out there looking for it, or you will have a nice conversation, and then you hopefully will be reminded in as much as maybe their topic or their view will change in the future, because that's also important. So I think it's pretty much like with big pharma. So they have their certain topics they are looking out for today, but that might change actually tomorrow or in a week. And they are restructuring basically their goals. And so for that reason, I think you have to be very open-minded. Yeah.
As a successful biotech entrepreneur and physician yourself, you've clearly seen like a lot of ups and downs. What exactly are the biggest pitfalls you've seen when academics try to translate their own discoveries into products?
Yeah, this is also a multi-level question. I think on the academic side, it is important if you think about translational medicine that you should, so this is just my personal perspective based on the experience, you should start thinking with the end product in mind. And this has to do with the fact that there are certain necessities. If you move a biological concept or maybe a mechanism that you think that is druggable, really into the clinic. So you have to closely look at the concept, or basically the realistic way this product would exist. So for example, in gene therapy, there are certain points that you certainly have to address immediately at the very beginning of the development. And normally these developments are not necessarily incentives that you have in the academic space, right? So for gene therapy, it is extremely important that it's not only effective, but it's also that it's manufacturable. And this is something that you would not immediately think about, because as a scientist, you're super happy if something works, especially in an in vivo model, right? And very often you just use one concentration or one dose. So this is what you will find in thousands of papers out there, right? But what you need is an understanding of the dose efficacy relationship. So you need to understand, is there a kind of a therapeutic window that you have to hit, especially? And also, is there a dose toxicity relationship? And if you just think about these two topics, this is nothing that you would necessarily get funding for, right? Because it would mean you have to expand your experiment size quite significantly. But nevertheless, so this is something that you have to do at the very beginning. And at the same time, you already have to think about manufacturability. So if the yield, for example, of a certain adeno-associated virus that you would pick for the therapy together with your transgene, if the production yield is very low, it would not necessarily matter in a basic science, I would say, environment. You just maybe do a couple more productions. But this would drive immensely the cost already if you think about the clinical applicability, right? So these are the things that you have to think about very early on. And it changed a little bit if you think about small molecules or maybe also cellular therapies. But it's always important that you look ahead, very far ahead, and ask yourself the questions, what would be these kinds of really denominators that also decides about the fate of a therapy. So that's something we are trying to do now. So we are always think with the basic science head on, but we are also trying to anticipate if we think about potential translatability in these terms.
And you've also mentioned costs as one of the biggest factors in this scenario. So obviously gene therapy is a very expensive procedure. How exactly would you suggest, or what are the ways you think would make gene therapy an equitable therapy method for heart failure patients worldwide?
Yeah, so maybe let me start at that point. So I think gene therapy by now is kind of an established concept now in the clinic. So you have seven EMA and FDA approved therapies for various indications, not yet for cardiac indications, but for example, Luxturna or Zolgensma. I think these are AAV2 or AAV9, various isoforms or serotypes of the adeno-associated vectors that are working in the clinic, but still having their difficulties in terms of safety and I would say in terms of how many patients you can actually treat with that. So since these are small numbers of patients, and as you have just mentioned, manufacturability is an issue here, the cost of goods, basically what it costs to produce yet or to date, these adeno-associated viruses actually also drives the cost of these therapies. And if you think about the numbers that are out there in terms of $500,000 for the treatment of one eye, or maybe another $1 million for the treatment of a patient with Zolgensma, I think this is for sure nothing that would be by far realistic if we think about a prevalent disease like heart failure. So what we are already looking for, and what we are also very actively discussing with our CDMO, is how to bring down costs. And this is something that is widely debated in the field. So certainly in terms of scalability, I think this will change things. The more successful therapies that will enter the market will certainly bring down costs in terms that the technology will enable larger volumes to produce at once. So this is certainly something that is not necessarily currently in our hands. So Aavigen is not a fully integrated preclinical stage company. We don't have our own production capacity, so we have reached out to a CDMO in Paris, SK Pharmateco, but this is a very active debate. So cost of goods have to come down, and I think it is, I think quite fair to expect that this actually will happen. But it will certainly take a couple of years until we are there where we would like to be. Yeah.
Could you explain for people who are new to the field how this works, gene therapy with viruses, because someone who doesn't know about that could think, oh, a virus, that's making me sick, right? So how does it work, a therapy like that?
Yeah. No, this is an excellent question. So yeah, of course, if we talk about viruses and you just think about what we all had to experience in the last couple years with Corona, this is certainly something that could scare people. Yeah. So what we talk about are viruses that do not have any longer the ability to replicate. So this is a kind of genetic engineering. We are talking about recombinant viruses. So if you look at the genome of a virus, you can identify various regions which encode for proteins that are necessary for the virus to replicate. And for the adeno-associated viruses, anyway, they need another virus. They need the adenovirus to replicate under, say, wild-type conditions or in the real world. But this ability has been taken away. And by that, you also free space. So you have now free space for your desired genome or your desired genes that you would like to put into this empty space. And at that stage, the virus just become a shell, like a taxi or something that can deliver genes like a football or a soccer ball you can think about. And then you put your gene of interest, your therapeutic gene. We talk about therapeutic payloads. You can put into that, and there are certain technologies then to amplify this virus, but whatever comes out of that, it has lost the ability to replicate. So from then on, it is just kind of a taxi that delivers the gene in the best case scenario into the organ or the tissue type that you would like to have. Then this virus will be unpacked. The genome will be released, and based on the biology of the virus, and in the case of AAV, most of this genome then becomes circularized, and it stays in the nucleus, is not integrated, and there it will be treated as just another gene that can or is being translated then into a protein. So you use from that stage on the host's or the cell's protein translation machinery, but you deliver the functional copy, for example, for a healthy gene, and you bring it in with the virus. So it's these two components.
How can I imagine the delivery of the drug itself? Is it something that we put in the blood, or is it a pill, or is it an injection directly in the heart?
Yeah, that's another excellent question. So if you look at the current gene therapies, for example, there are, I would say, two options or two opportunities out there. So Locturna, for example, which is a gene therapy that treats a hereditary kind of blindness that's directly injected into the eye, into the subretinal space, because it makes a lot of sense to do it locally, because you can also use a low dosage. If we talk about Zolgensma, for example, who is targeting a motor neuron defect, so you have to bring this somehow into the central nervous system. And here, one approach is to inject it intravenously. And this AAV has this ability to cross the blood-brain barrier, which not all AAVs can do. Nevertheless, you have to use an immense amount of virus to bring a smaller amount over this barrier. And then Zolgensma actually can get to work. But you can also inject it intrathetically, meaning into the liquor space. By that, you can also, decrease the amount of virus, but you become more invasive as a simple intravenous injection. So these are, I would say, the very first generation or natural recombinant AAV serotypes. So what we are working on are organ-targeted AAVs, and this happens in a number of other indications too. So people are looking for brain, even neuron subspecialty targeted AAVs. So we are doing this for the heart, because the concept of Aavigen is to inject these cardiac targeted or heart targeted viruses that we are developing intravenously. So in a best case scenario is that a general practitioner could at the end of the day inject this therapy with a short infusion, maybe 15 minutes. through the peripheral vein. Then the virus circulates through the bloodstream and it targets the heart, but avoids all the other organs that we would like to avoid to minimize toxicity. So that would be the concept there.
How does it manage to avoid the other organs?
Yeah, so this has to do with the way how we are developing this. But and if I say we, so now this is the team in Aavigen. So we have 10 young, really super ambitious and talented people in our research team. And then Dr. Martin Busch, who is one of a former colleague from my division. So he made the decision to transition to 100% in the company. So he's also one of the co-founders. Martin is also heading the team. So what we have done is we have established a pipeline to genetically engineer these viruses in as much that we can change the composition and the structure of the capsid, so the outer shell. And the outer shell has protrusions and loops, and with these molecular fingers, basically, it can interact with the various parts in the body that you need to interact to bring something specifically to the heart, but also to avoid interactions, right? So with other parts, for example, not bringing something into the liver. And the way we have set up this pipeline, we have a number of in vivo organisms where we are working in. We also have, which is super important for us, an ongoing collaboration with the German Primate Center in Göttingen, because at that stage you would like to get as close to a human as it is possible. And non-human primates are at that stage the closest organism that you can actually utilize for the benefit of the patient at the very end. And so we are generating libraries that contain millions of these variants, and they are done more or less by a shuffling process. So you chop down basically the genome of the AAV, and you mix it, and by homologous recombination, they assemble back, and then you have, as I said, millions of different variants. So you are then basically injecting them intravenously, and you pick those at the very end through various iterations that always make their way in the competition against each other to the heart. And then you also negatively select against those who are ending up, for example, in the liver or in other organs that would drive toxicity. And with this kind of selection process, you get to a certain group or to a certain portfolio of those viruses that can actually deliver what you would like to see.
And now you've also mentioned like several of the processes through which you have to select the gene of interest and then insert it into the viral vector and so on. So a tool that is obviously widely used nowadays is, of course, artificial intelligence.
Yeah.
And how would you say, or rather, would you say artificial intelligence makes your work a lot easier, or does it assist you in any way? And do you think that maybe one day in the future, artificial intelligence would be able to take over the role of humans in gene therapy as a whole?
Also, excellent question. I think it's not an easy answer to that. Yes, we are already using these tools. We have experts in our company who are trained in that. And there are certain aspects where you can already apply it because you understand the environment and you understand the capabilities of the technology. And there are other areas where I think it is probably still very ambitious to apply it. We have also reached out to a number of different companies who specialize in AI. And our current understanding is, as I said, there are certain stages where you can use it. For example, in using the data that we have already generated now from our screens. So these are excellent training data, which an AI algorithm would need in terms of what are the accepted pools going into the heart, what are the rejected pools not going into the heart, into the other organs. So by that, we are refining constantly the pipeline that we have built. And this is something probably that exceeds a little bit our podcast here, but it's not as simple as you just put a virus into the organism and then it binds to a certain target on a cell. You have serum components which are basically jumping onto the virus So they are more or less opsonizing or coating the surface. So you're creating new epitopes there. Then you have to cross the endothelial barrier. By that, you lose a lot of this virus, and then you end up into the interstitial space. And there you have a number of matricellular proteins where, again, the virus is binding to. And then as a third stage, it sees the cell or it sees the target cell. And by there, you don't really know what's sitting on the virus in terms of additional proteins, new epitopes. So if you do this in vivo screen, you don't care at that point because you just look for the winners, right, who made it through. But applying AI to this process where you actually have no understanding at all which are the necessary key intersections, that turned out right now that it's not yet working. But I'm pretty sure it will work maybe in a year or from 2 years right now. And we are on to that because I think this is really key because it can speed up development quite significantly. Yeah.
How does the business model look like? Are you developing a platform that you could then sell to any other companies who want to bring a therapy to the hearts?
Mm-hmm.
Or would you only use the therapies you develop in Aavigen?
Yeah, I think if you are on the bio-industrial or entrepreneur side, you have to be very flexible. So I think you have already mentioned the opportunities that are out there. I think once we made the decision that we would move away conceptually from the basic concept of the first startup and not necessarily using a catheter-based local administration, which has pros and cons, basically. We had to develop these viruses because they were not there yet. Conceptually, I think, or technically it was possible, but not really many people took on this advantage because it takes a while to do this. And so the idea is really to develop our own lead therapeutic products and chaperone them as long as possible until they are in the clinic. And this is maybe a lesson learned from our first startup. There, I think we were not yet as experienced as we are today. And I think we certainly gave up a little bit too early, I would say, the final saying, what happens at which stage. So we were still instrumental to the development. But we have learned that if you work with big pharmaceutical industries, so we had the privilege to work with Bristol Myers Squibb at the end, there are certain elements within these companies that can even crush a very robust therapeutic concept against the wall. And that's something that we've learned at that time, which was important. So we are leveraging this knowledge right now. And so our concept is to keep basically the development of our lead product in our hands as long as possible because we have deep medical expertise in the company. So Professor Hugo Katos, who is my, I would say, key European mentor on that side, he is on board. He's our chief medical officer. So I think we have a very deep understanding about the disease, the medical indications. And so for that reason, we are quite sure that we can move this forward into the right target population. And then, as you have mentioned, if we are talking about a distribution of the product into the European Union or maybe also globally, you can't do this as a startup anymore. So you have to seek strategic partners very early on. And then we are also talking about the opportunity of licensing some of the capsids that we do not need. So we have more capsids at hand that we actually can use for our therapies. And there we are also actively seeking licensing collaborations in indications, which is a nice word, which are not cannibalizing basically our own lead indication. Yeah.
So, of course, gene therapy has been quite the controversial topic over the past years. What misconception are there any misconceptions that you would kind of like to clear up about using gene therapy as a solution towards a cure?
Yeah, I think it is a little bit as with many other therapies. I think if you look, for example, at I would say the whole antibody space today, antibodies, especially in the area of immunological diseases, I think They had seen their ups and downs, I think, throughout almost 2 decades. Now, today, it is something that is absolutely a basic foundation in clinical therapy. And I think the expectations for gene therapy, and this is, they're always high at the beginning. And I think they are not realistic, right? Especially when it comes out of the academia. So there is always this tendency that things are super hyped. For example, as CRISPR-Cas technology, gene editing. It's a very super elegant tool, of course, but to recognize the biological complexity that you are moving into, right? So there are certain things that you can't really recognize at the beginning. So you will always have pitfalls. But if you think about gene therapy entering then the clinical stage, you have investors, you have market expectations, basically. And so here you have, I would say, an environment which is, as I said, on the one day it's extremely hyped, on the other day people are super disappointed. And this is not really driven by a realistic scenario. It's rather than this melange of interest in, return of investments and get this very quickly, but on the other hand, understanding the necessity for long-term development. So I think what we see is something that in gene therapy right now, what we have seen in other drug classes too. And I think this will certainly flatten out in the future when we get more therapies into the clinic.
You're talking about this long-term aspect. Biotech is an extremely long-term business. It's not, I start something and 1 year later,
No,
I'm good, I'm done, I sell the company.
Yes.
How do you manage the frustration and the patience that you need to go through basically decades of leading a company? What motivates you in the day-to-day? Is it like the end goal, curing patients, or is it the intrinsic things you do every day that just feel good and that keep you going?
Again, I think there are certainly a number of answers that I would need to give here. I think first of all, working in the company, as you have said, of course, I think there are everyday challenges. But I think this is, kind of the rewarding aspect. So you have to constantly adapt your strategy. So I think if we sit down, we have, for example, leadership team meeting every Friday. And if we get off the table and think, now we have a good strategy for moving forward, the next day you hear some news. And I think you're aware what's currently happening in the world. So the market is changing dramatically in terms of where investor money is shifting to. And then you realize on Sunday, we might have to think about what we actually do in the next couple of months. And yes, I think it is this kind, I would say, of rewarding effort that you have when you see that things play out. And on the other hand, it is really that you understand it's not really a frustration, I would say. It's just that things do not necessarily work out along the expectations that you had. And then you have to adapt, right? And I think what's really nice is that you can combine this with working with this super ambitious team that we have. And I think this is something where you get the energy, right? Also from sometimes you have things that do not really work out strategically, but on the scientific level, you really have kind of a small breakthrough. And then you have to take your energy from that, right? So I think this is the way how you do it. And in terms of if you look ahead, and this is still something that motivates myself, I think I can only speak for myself, but I'm pretty sure I also speak for Hugo and for the rest. It's really bringing something to the patient where we are absolutely convinced of that it will be a game changer. Yeah, so I think this really drives us.
Mm-hmm. Now, of course, in business, it is a very risky field. What exactly would you say was the biggest scientific or business-related gamble you've taken so far? And what exactly has this experience taught you?
Yeah, I think it is the experience that we made with our first startup. So I think we were quite happy when we got together with Unicure. And I personally have to say I have learned a lot. So I've met great people, for example, Jörn Aldag, the CEO, who took me literally under his wings during that time. So through him, just to mention one aspect, I've met on a very regular basis with high-profile investors in the US, with all the banks you normally hear throughout the day if you listen to the news, and really learn how to deal with these people, understand conceptually and strategically how this business at the end of the day works, and what are the potential no-goes and goes if you are moving into that area. And another aspect is at that point then that when we got together, for example, with this bigger pharmaceutical company just mentioned, we learned also how risk-averse these teams actually operate, right? And so we just published a paper. It's about, again, S100A1, about this particular protein we're working with. And we used another serotype. It's called AAV5. And we showed that it has a very robust therapeutic effect in these disease models that we also used back there. So it works quite nicely. But it didn't work in the hand of the CRO and the CROs that came into play when we had to work back then together with Bristol Myers Squibb, and they became nervous and really didn't understand. And we told them it's not the therapy, it's the CRO who is the problem here, because you have to be properly trained in this catheter-based technology. And as we have just shown now in this double-blinded study, it works perfectly. So unfortunately, at that point, we realized that we will certainly not going forward. And we also made decisions then to part way. So our first startup definitely was a success in terms of exiting, or having an exit with a company, but we didn't made it to the finish line. We didn't bring the program into the clinic. But with all that I had the privilege to learn at that point, it was kind of a logic decision to think about the second startup, but then leverage the more advanced AAV technology, and bringing this forward into this new therapeutic concept. Yeah. But also let me say, because this is also super important, we are, I think, embedded here in an academic environment, and it only works if the different, I would say, parties really have an understanding of what it is needed that something like this can take place. So the university and the university hospital were super supportive with the first startup, especially our former rector, Professor Eitel. So he was super instrumental in bringing basically the opportunity for me to work in the first startup, and also now with the second startup, we had the opportunity to find really, I would say, good terms with a university hospital. So I think it reflects positively back on what an academic institution can do, right? And if you think what's happening, for example, now in Baden-Württemberg in the land in terms of the major industries are currently undergoing, right? I think startups are not just something that happens in the kindergarten. It could be really something if you look to Mainz, for example, with Biotech. So Ugur started also very small, but now they're a major player, right? And I think this is something that I'm not high-nosed now a claim for us, but I think it is really the opportunity that we have here on the campus, especially because we have high-profile science going on, and I think there are really many opportunities on the table.
How do you manage both leading a company and being a professor? What does your day-to-day look like?
What does my day-to-day look like? Yeah. Again, this is a team effort. So I'm super privileged on the academic side to work together with Dr. Julia Ritterhoff. Julia is co-heading the lab with me, because without Julia, it wouldn't be possible. So you can't be at the same place at once, right? So it's not like quantum mechanics where you have the different spins. No, and so Julia is doing a lot of the work in the lab. So I have the opportunity to arrange my time quite as it is needed, right? So I have certain times where I'm here, which are the team knows I'm here, and especially working with the leadership team, and then our fixed times in the lab, and then I have days in between where I really know I either have to travel or I have to do on a very short notice some things that actually I take care of. But it wouldn't work without the co-leadership on the academic side, and basically the co-leadership in the company, right? because we are 4 people making coherent decisions, and then you have to bring that together. But and I think this is also, I would say, the beauty of the work. You are not always certain what you're doing next week, right? So it could be something completely different as you had planned or expected, because you have to adapt to what is necessary.
Of course, as you've mentioned before, Heidelberg University is a place where a lot of high-profile science is taking place. So for young scientists with a therapy idea, would you advise incubating it within academia itself, or would you advise kind of like spinning the idea out early into the industry?
Yeah, excellent question. I think we have debated that question with a number of younger colleagues a couple times before. I think what you have to take into account is that inside that question, there is a kind of an internal conflict built in, right? So as a younger scientist, first of all, you are not, I would say, completely free in your decisions, what you are doing with whatever you are researching, right? So you have your supervisor. First of all, you have to align with that person. In the best case scenario, you are aligned that whatever you might find, if it has a kind of a translational value to it, so you would be able to move that forward. But I think at that stage, it is important that what lures a little bit in the future is already the question of patentability, right? So you have to somehow, at some point in time, you have to secure the intellectual property that might lie within this development or discovery. And this contradicts a little bit of your interest to build your career on the necessary incentive that you have to bring forward as a scientist, which are publications or presenting something at a scientific meeting. If you do that, you disclose the information. From then on, it's public, and then it's not patentable anymore, right? So you have to find a balance for that in a best case scenario, and I think we have that. You have a technology transfer office who can advise you how to move forward, because then you can hopefully find a good compromise between still focusing on a publication that you would need, but on the other hand, secure the IP also in a substantial way, because you need also strong data for the patent. And if you can manage that and you get the advice, and I think our institutions are built around this concept, yes, then both is possible. Sometimes it works out, sometimes it's not working out. So, but this is, I would say, a numbers game. Yeah.
Is your main job as a CEO to be the one looking very far into the future and see which problems might come to the company and be the first planning on that? Is that the substantial time of your CEO?
I would not, so I would not claim for me that I always have the best ideas. No, but it is, I think I would really say it is this job. Yeah, so you have to trust your integrative, how do you say, quote unquote gut feeling, which is probably a non-conscious integration of all the various things you have learned throughout your stage at that point. Yes, so this is something that especially I'm focusing on. So trying to understand what would be necessary adaptations, and then bring this concept into the leadership team, discussing pros and cons, and then also discuss that with our investor and align this, and to make sure that at the end of the day, if you make decisions, that you are still within the project development timeframe that you need to keep. But it's extremely important to be out there and to understand what the various levels are that are affecting your development. Yes. Yeah.
Is a big part of it to travel, meet people, and get other people's point of view to see what you don't know that you don't know?
Yeah, exactly. I think this is, I think you have pinpointed it. It is extremely dangerous to be high-noised or arrogant and think, I'm pretty sure that this is the right way. So it is exactly, so you have to listen, be very open-minded, and try to build scenarios. So a lot of the things that we are doing, and here, for example, Mark Lerchenmüller comes into play, because I think he has a very proficient Boston Consulting training under his belt. So we are building scenarios, and we are trying then to put basically risk numbers to the various scenarios, and then make decisions and understand if you would choose a certain path, how this would affect basically another scenario. So you always have to have a plan B and C, and this is something we are trying to operate. And I think so far we have navigated this little ship through, I would say, a number of rough sea conditions. And but being open-minded and listening, and being flexible, I think this is a super important task.
How did you meet Mark?
Actually, this is a funny story. We've met through his wife. So his wife was one of the doctoral students that I supervised during my time in the United States. So in 2006, my US-based mentor, Professor Walter Koch, after I had my postdoc time with him, he made me an offer for professorship, and I was quite flattered, but still said, this is kind of a move. I finished my clinical education then in Germany, but then actually, and maybe this is another advice, if you're young at that stage, take the risk because you have the opportunity. So I made the decision to leave Germany and take on this position in the US. So Mark's wife, she heard about that, and since Mark was going to the US at that time, she was looking for an opportunity to follow him. They were not married at that time, but they were together. And so she came up to my office when we were still in the Otto Meyerhof Zentrum, where our lab was located. And so she said, look, I'm looking for an opportunity to do my medical thesis in the US. And then we arranged everything with the medical faculty here that would be possible. So we've met through her. And then I've met him in Philadelphia. And so there the story started actually. And since then we became good friends, but also business partners today. Yeah.
What's the criteria that you see in other business partners? At which point do you think, okay, this is going to work? Let's do it.
You mean in terms of in the team, or
in terms of the person, the trust level, the shared vision? How do you select a co-founder?
Ooh, that's a pretty good question. Yes, of course. So I think, as you have said, I think you have to have a certain understanding of the common goal, right? And I think it is somehow that it's like if you are in a boat and you have to row. Sometimes, I don't know, one of the persons have to put in more effort, and then sometimes the other person has to put more effort. And we made very clear that the team is equally incentivized. So that means that whoever is for some time putting more effort into the thing, he knows that at a certain other time, another person has to be, I say, more active for certain parts, because we have this division, right? So Mark is taking care of the economic part, and Hugo is doing the medical part, as you have just said. So I'm working on the strategic part, but I'm also integrated in all the other things. And Martin is pretty much taking care of the team and of the operative side. So everyone has basically its high time and it's rather than maybe lower time, and we are looking out for each other. But you never know at the beginning if it will work, right? So we also had, of course, kind of important discussions where we had to balance out certain opinions, and by that you learn each other. And I think we have really built a pretty strong team by that. But I think knowing each other helps a lot. And I think at that point, we differ from maybe other startups where you buy in the CEO and you bring in people who are on that side for quite a long time in the industry. Of course, if we are moving Aavigen ahead, I think it's not realistic that we will stay always on board in the functions that we have right now. I think we might have to transition also to other positions then. But maybe that's the best answer I can give so far to that. Yeah.
Now, was there ever a period of time, or point in time rather, where you thought that a project was about to fail, but then you sort of pivoted that and turned things around for the better?
Yeah, I think Corona was extremely difficult. Yes, because we had our investor on board, but basically we couldn't start. Yes, because it was not possible really to work. So it took us a while, and I think that was probably not a bad thing to carefully select people. So our very first scientists that came on board, they are super instrumental for what we are doing today. And the selection of that team was extremely important. And of course, like Winston Churchill said, right, never give up. Because if you give up, you have already lost. So at that point, you hang on, right? And sometimes, as I said, you have to create opportunities, meaning that you have to create several scenarios. And if scenario 1 is not working out, then hang on to scenario 2, right? And push that forward. And it's, so you have to be perseverant and basically not giving up. And I think at that point you already differ or distinguish yourself from a lot of other people. And you have to go through these downs. But so far I think we were lucky in as much that we managed to create the necessary scenarios to move forward. Yeah.
Right now, funding is scarce for biotech companies. We're recording the episode in the fall of 2025.
Yeah.
How do you feel about the next years? Do you feel it might be a biotech spring again, or do you think winter will last for a bit still?
Yeah, pretty good question. I think what you can learn, and again, this is my personal perspective, the way how this whole investment field also works is that you have to find a lead investor. And normally this lead investor will do his due diligence, meaning it will look in whatever you have created, will search through the data that you have if everything you bring to the table is substantiated. Once a decision has been made there, you have a number of VCs who are then jumping on the wagon, right? So for that reason, I think it is a process that is understandable, and I think you have at that point, to be consistent enough and perseverant enough to talk to investors, build relationships, even if it's not happening today. As I said, their interest might have changed tomorrow or the week after. What we see right now, I think, is a big movement of away from biotech into AI. I think which has a number of reasons, geopolitically motivated, and that, as you have just said, doesn't make it easier for biotech. But still, there are substantial funding rounds out there. I think money is there, but not necessarily, I would say, not in this high numbers as we have seen it in the past. And again, here you have to adapt, and we are trying to do this. So we will be at Jefferies in November in London, and again, talking to investors. And as I said, building relationships and being professional about what you are doing and what you are presenting, and highlighting the opportunities out there. And don't be frustrated. This is what you have to learn about. Yeah.
When is the best time to start building those relationships with investors, to start this, I'll call it networking efforts for students, for example? Do you advise them to do it as early as possible, or just later when they want to go into biotech?
Yeah, I think also I would say, if we talk about the investors, I think you have, of course, these are organizations with different levels, and until, I would say, also for us, it's always important to talk to people who actually have decision-making capabilities. But maybe let's turn around. I think if you are a younger colleague, I would advise them maybe also ring our bell. So if you are in summer break or winter break, maybe ask if you can look over our shoulders for a couple of weeks, and so you would learn how a startup works internally. We had this with our first startup, so we had a number of young academic colleagues coming on board, and they, for example, made it clear to us they had applied before, whatever, to other companies out there, Roche, Boehringer, and they were always saying, you are lack bioindustrial experience. I say, of course, because I'm applying. I'm applying at your door, and so after then, we exited basically our relationship. Of course, we also had to shut down our Heidelberg Unicure Germany partner site, but these colleagues, they quickly found after that, they immediately found new jobs in bioindustry. So they collected this needed experience very early on. And again, it was a super interesting time. You have young colleagues. You are capable of also taking some risks in terms of what you develop in the company. I think we enjoyed that from the leadership part at that time. And they really, I think, had the benefit that they could move on very quickly. So that was a nice experience also to see that this, I think, came together, right? But you also can make the decision, stay in academia for quite a while, and then, for example, try to apply and ask companies if you can be there for a couple of weeks. I think we have a number of startups here in Heidelberg who are certainly open to that. Yeah.
Now, if you were to give a single piece of advice to a master's or a PhD student in terms of them wanting to blend the fields of both science as well as entrepreneurship, what would that be?
Yeah, I think it might be something like this to start with. So look, if it's possible, look into a startup, right? Go there, maybe even knock at the door. Say, look, I'm here. I would like to understand how this works, how this operates. And this is probably a real world experience. You can also, I think, apply by now to certain courses here at Heidelberg. Which are, I think, concerned with the task to bringing these two different world experience to the younger students. So they have engaged in speakers. So, for example, the German Cardiovascular Research Center is also doing this. And from time to time, I'm invited giving a lecture there. So you can hear basically what people have to say who have done this. Yeah. All theory is gray. So I think it's the best thing you jump into the matter and you knock on doors and just really make the decision. I'm looking into this startup field and work there for a while, maybe a couple of weeks, and then have probably a much better understanding because then you can talk to the employees, you can talk to the people. And as I said, we would be open to do that. Of course, we can't take in 100 people, but I think we would be very open to do things like that. Yeah.
How do you feel that the entrepreneurship part of your life changes your professorship part of life? Do you feel it inspires you? Do you feel it brings a perspective that makes you a better professor?
Yeah, in terms of what we do with a translational perspective on the academic side, you certainly benefit from being on the bioindustrial part, on the entrepreneurship part, because it sharpens the thinking, the way you make judgments about what is potentially translatable or not. And for sure, this is not always a 100% decision. You are not right or wrong, but you can put certain, I would say, likelihoods to it, right? Certain percentages. So I think this certainly is something that benefits there. And on the other hand, in terms of the academic thinking, which I think is this incentive that you can be creative, right? So you still have this kind of degree of freedom and put it into the company, and then you have to fight with the team. So they hold you back in terms of, so now this will completely blow up our timelines, or you think, this is absolutely necessary in terms of conceptual changes. But what is important is that also that we have, which is good, we have a clear separation between what we're doing in the company and what we are doing in the lab. So there is no crossover in terms of ideas, but conceptually the way how you maneuver on the academic part, in terms of how you approach a problem, right? It's a way of thinking to, look from every side. I think this is important, that you still keep this also in the bioindustrial world, and then on the other side, that you still have the curiosity to look after new basic science principles, and not, how you say, always ask the question, is this translatable or not? That's not the key point, but at a certain stage in time, when you have developed something new, or you think, this looks interesting, then you have the advantage of applying this kind of thinking. Yes, so I think it nourishes each other. Yeah.
You also tried out both sides of the Atlantic. So you were in the US for a while. What differences did you notice in being a professor in the US versus in Germany?
Yeah, I think when it comes to, I think quite an important aspect is funding. So NHLBI funding was, I can't speak for today, because I think they have tremendously cut down the indirect costs. But funding, I think, is much more substantial in the US. So they have certain instruments, funding instruments, that if you compete for that, and then if you then are successful in receiving these grants, you really have substantial funding. And also the degree of freedom, the way how you operate with the money, is much more flexible than here. So for example, we had a business manager for our unit. And as funny as it sounds, I had a credit card for my accounts. And whenever I had a problem, so I had to build a special device, right, for whatever, blotting proteins or something like that. So I went there and said, look, I need the permission to go to a certain company in Philadelphia because they can build me this. It's a 5-minute decision. And you can do it. So you're super flexible and you're super effective. Here, it will take you through quite substantial paperwork to do this, if you're allowed at all. So I think there is, I would say, higher flexibility and higher funding. I think, as you have certainly heard, conditions have changed. I think right now it's much harder. But I think also the level of competitiveness is higher. It forces you to be super effective and invest your time quite efficiently. I absolutely do not regret that I have done that. And what I haven't mentioned, and that was also kind of a super interesting scenario, because at the time when I made the decision that I'm going to the States, of course, Hugo Cartus, we intensively discussed that, because I also wanted to hear his opinion. And he said, look, why don't you keep your lab in Germany? And so I've kept the lab in Germany, actually. And then going also back and forth from the United States, and with the support of Hugo, that was possible to keep up the lab here, and really never lost track with Heidelberg. And then at a certain stage of time, this opportunity basically opened up here. And so I made the decision to come back then, because I think at that point I have learned what I wanted to learn in the US, and it was interesting to take on this opportunity here. Yeah.
Now, if you were to go back in time and then look at your career, would there be anything that you would do differently, or would there have been some things that you would have completely avoided altogether?
Oh, that's a good question. I think, in general, if I look back, I think there is certainly one thing that is important. If you make decisions, you have to accept the consequences, period. And if you don't like the consequence, of course, you can start to fiddle around and be uncomfortable or whatsoever. And I think this is something that I have never done personally. So if I had made a decision, I stuck to it. And I think this is also something that is important. I think you have to trust the work that you are doing, and especially when it comes to the question why we are still doing this, what we are doing today, because you are creating these stepping stones, right? And if you have a solid foundation, if you have moved forward and the next step fails, you can always jump back and you are on safe ground. And this is the way how we operate, irrespective of whatever is asked for you in terms of be innovative or whatever. You have to go forward with whatever you do. So this is extremely important, and this is also what we teach our students, right? So if you do science, you always have to have a clear foundation of data sets which are absolutely solid. And from there you can take risks, right? But don't do a leap of faith and do 2 or 3 jumps, because then if you fail, you end up with nothing. And I think this is important. And if you do that, you create this, I would say, this red line, or this trajectory that your path is following. And this is something that I have tried to do all my life, and so far it worked out. And this is maybe some kind of advice that I can give. Trust yourself, but on the same time be very critical to what you're doing. But if you are sure that the data are the data, trust them.
Do you dare to give a timeline of when you hope to see gene therapies in patients curing heart diseases?
Yeah, so we have an ambitious timeline, for sure. I'm saying ambitious because with the changes in the market conditions, I think it's certainly still ambitious, but we stick to it. Yeah, so we are planning for end of 2027, actually, to move into the first clinical trial. We are working intensively on the way how this trial should look. Also, we are debating intensively where it should take place, because some of the places that we planned maybe earlier this year might not be so attractive anymore in the future. So there are some decisions that we have to make. But also here we have a number of partners we can count on and work on. But end of 2027 currently. This is kind of a timeline that we have given us, and so far everything is geared towards this point.
And then 27 first clinical trial. There's multiple rounds of clinical trials, the phases that have been that need to be completed. Assuming everything goes
according to plan,
somewhat right. What's the soonest it could be in the clinics used on patients?
No, I think this is the date. So if you are entering the first clinical trial,
okay.
So I think in the gene therapy field, you have the opportunity to use kind of, I would say, innovative trial design.
Mm-hmm.
So we would certainly think about kind of what we say an adaptive trial design. So we would, for sure, safety is the key issue, but you would like to build in very clear signs of efficacy. So this is something that we have in mind, or what we have already mapped out. So this would already mean that you would treat a group of patients from, I would say, 50 to 60 patients onward. So that would be the first time what we call a gene therapy medicinal product would be in the clinic. And after that, of course, then you have additional stages where you then enlarge the patient groups, because then you have to focus more on efficacy, with the idea, of course, then bringing forward very early on a pivotal trial, from where on what we call apply for a biological license application. So you can bring it basically already into a marketing process and still work in late clinical development, but the drug would already be available at that point. Yeah.
Is there last points you would like our listeners to have from the conversation?
First of all, thank you for your interest, and of course the opportunity to reach out to I think super interesting opportunity, I think what in terms of, because you've also mentioned the wording misconception, because what I've been asked from time to time, of course, in the lab we are actively looking for PhD and medical students, because I'm often approached being asked, are you still looking for students because you are in the company? Of course. So we are open there. We are also looking for colleagues from the clinical department. So on the academic side, really nothing has changed. We are super open. But I think what we can offer is really this perspective, right? Looking onto the other side in terms of bioindustry and providing insights, and also providing opportunities to understand very early on how this would work. I think this is maybe just making some advertisement for the lab here, if that's okay.
Of course. Patrick Most, thank you so much for taking the time to do this episode with us. It's been a pleasure talking to you, and we hope you enjoyed it as well. We'll see you listeners to the next episode of our podcast. Have a good one.